go to Gary Michelson for this generosity. We're at something of a turning point in the careers of the individuals and possibly also in the epidemic, which has slowed down a lot of people's research and a lot of work, just simply not able to get to laboratories and so on. And so it makes this year's round all the more exciting, all the more extraordinary that these young people have been able to do so much in the time of COVID. Our next round, by the way, will open for nominations on April 1st. So make a note as you watch this, if you're considering the possibility of applying for the 5th round of Gary Michelson Prizes. This is, of course, also brought to you by the Human Vaccines Project, which has for many years been seeking to find truly spectacular innovations in immunology and in our understanding of how the human immune system works, literally trying to map the entire human immune system to know how it responds to disease and causes disease, both of which we will be talking about today in the context of these fantastic prizes. And I want to first begin by introducing you to the man who runs the Human Vaccines Project, Dr. Wayne Koff, who's been in the business of searching for vaccines for 4 decades now, once at the NIH, at the Human Vaccines Project, the International AIDS Vaccine Initiative, and now running the Human Vaccines Project. Wayne, over to you.
I'm much younger than that, Laurie. I don't think I've been doing this for 4 decades, but it does seem like a long time. Well, listen, it's wonderful to be here. This is one of the best of the days of the years for us, this was an unbelievably complex, year, and we got over 150 applications in this round of the proposals. And I just want to congratulate all of the winners. They are exceptional scientists, and you'll see in a minute the interesting proposals that they have. Obviously, that there wouldn't be a Michelson Prize if not for the incredible amount of the vision and generosity of Ollie and Gary Michelson. And so I'm not going to make a laudatory introduction. Gary, I know
Thank you.
He wouldn't want that. And so I want to go ahead and just go ahead and introduce Gary and thank him again for being an incredible partner with us on the journey here of the Michelson Prizes. So I'll turn it over to you, Gary.
Thank you, Wayne. There's an old saying that one can't leap a chasm in several small steps. And while most science is incremental, I believe that great science leaps. So what does it take to do great science? And I think 5 things. It requires intellect, knowledge, imagination, courage, and perseverance. So why courage? Well, first, because this type of research either pushes past the known limits or attempts to disprove the orthodoxy of the moment. And of course, there's a substantial likelihood that it will not succeed. Now, not succeeding is not the same as failing unless that is where you stop. Now, I see these qualities not exclusively in young people, but very much in our young scientists. These young researcher prizes have succeeded beyond my expectations because apart from the research itself, the prizes have markedly accelerated the career paths of each of the recipients. And that's just been a blessing. I'd like to thank Wayne and the Human Vaccines Project for all of their work. This could not have been done without them. It's a great relationship we have, and thank you so much. I'd like to congratulate this year's winners. And I would just say, as a final remark, if the past is prelude, then the 3 of you should really buckle up for what's going to be a fabulous ride. Thank you.
And thank you again, Gary, for your generosity and your comments. I'm going to take us on a short path through a major conundrum we currently face in the COVID fight that has implications writ large for our understanding of human immunology, and show you some interesting findings that perhaps will trigger some thoughts for next year's applicants for the Michelson Prize. We're on a journey that involves long COVID, and in particular involves what is going on in the brain and how the SARS-CoV-2 virus or the immune response to the virus is affecting the capacities and functioning of the brain. This was noticed from the very beginning. Dr. Anthony Fauci in 2020 was talking about this strange phenomenon referred to as brain fog, one of the many long symptoms suffered by people who have survived COVID, but continue to have lingering symptoms for, we don't know how long, frankly. It could be, in some cases, horrible to imagine that we're seeing permanent changes. Certainly, we've seen that the virus is expressed in the cerebellum. It can be found in various parts of the brain on autopsy and is clearly colonizing and reproducing in the brain. Therefore, has obviously crossed the blood-brain barrier. Some of the symptoms early on, noted 2 years ago with SARS-CoV-2 infection, came to be called long COVID, and they included mostly symptoms that are associated with mental health, fatigue, brain fog, sleep disorders, periodic fevers, anxiety, depression, nervousness, a whole host of different symptoms, all basically pretty vague, hard to pin down in clear diagnostic terms. They overlap with, of course, symptoms felt elsewhere in the body and that affect the cardiovascular system and the pulmonary system. Some of the clearest ones is this loss of sense of taste and smell. And less clear are these sort of brain fog, loss of concentration, the sense that you're pushing through some kind of mud in your brain just to pull your thoughts together and to follow what someone is saying to you. The path to recovery can be long, and in some cases, we have not yet seen full recovery in key individuals from this list of long COVID effects. And this means that the priority is there to understand especially what is going on in the brain. By introducing sophisticated brain scan technology to see what is actually happening in the brains of people, both during acute COVID and for the months thereafter, some interesting things are beginning to come to light. But before we get there with the details, we need to understand that the epidemiology of this is extremely complicated. And it's been up to now, I would say, really impossible to separate out the multitude of factors that could contribute to brain fog, to fatigue, and frankly, to death. And keep in mind that the majority of the people who have been dying from COVID have been seniors, people over 65 years of age. And that means that they already have a whole plethora of pre-existing conditions that may play into the picture in trying to understand what's going on with these long-term effects. And we may also see simple exacerbation of latent or pre-existing problems in the brains. That complicates things. In addition, we see that those who already had dementia or Alzheimer's diagnoses, if they were in nursing homes or were on Medicare, in large-scale surveys, turn out to have had their symptoms and their likelihood of demise greatly enhanced during the COVID pandemic. And here again, we have to ask ourselves how much of this may be because of viral infection. And you see markedly different rates of Alzheimer's complications between those who were diagnosed with COVID versus those who were not. But also the nursing home experience itself was already perhaps not a particularly pleasant experience. Add to that at the height of our COVID epidemic in the United States, we shut down access. Families could not be with their loved ones. Patients were isolated. And isolation, of course, contributes to all the negative symptoms of Alzheimer's and other forms of dementia. And this has further complicated everything about trying to understand long COVID and in particular, the neurological symptoms. We just don't have good definitions. Steve Deeks was speaking recently in one of the Human Vaccine Project's routine webinars and addressed this very strongly, saying that until we can really pin down some consensus diagnoses so that we know what we're talking about and have methods for clear measurement that are agreed across the board, all of this is going to be very difficult to fully understand, much less treat. And there are incentives for getting to the bottom of this because it looks like a lot of what we're seeing in the brains of people with long COVID are similar to contributors that we would call aging brains or brains, frankly, leading into dementia. And this may help us better understand those syndromes. So, for example, we see a huge increase in all forms of mental illness in young people across the board. Meta-analysis combing through multiple studies, 29 studies, more than 80,000 young people involved. You see an increase during COVID, 25% depression, 20% anxiety. And this is a problem seen all over the world. Now, is this because they had COVID, or is this because they're isolated, because they're not going to school, because they're having to spend so much time with their parents and siblings? You know, so many factors are involved in trying to appreciate why these mental syndromes should be in play. And this is, of course, also true across the board for adults. We've seen a nearly 28% increase in major depressive disorder worldwide, and a nearly 26% increase in anxiety disorders. So again, this is complicated. And is this correlation or causation? Are we looking at the virus causing syndromes, or are these larger societal issues? For example, this study out of Boston shows that a great deal of the depression and anxiety was associated in the United States with financial stress. And you see a class differential based on the ability of people to be resilient financially during the COVID lockdowns in terms of whether or not they express depression and recover from it. So this suggests that we're not looking at a pathological process, but a psychosocial process. But of course, that doesn't take care of all of it. Studies in the UK have shown a tremendous increase in mental illness, a frank diagnosis of 11% escalation amongst those who have been diagnosed with COVID. And brain scans have indeed found changes taking place inside the brains of people who have even had mild cases. We'll start with what we know now out of Wuhan. We're now 2 years out from most of these individuals having suffered from COVID in the original outbreak in China. And long-term study of thousands of these individuals shows cognitive impairment more than a year out. Now we're 2 years out with some of this data. Long-term cognitive impairments in more than 12% of the individuals, and real cognitive declines. Sorry about that phone. We're seeing a clear demonstration of peripheral neuropathy with actual nerve damage in a fair percentage of individuals who have been diagnosed with COVID. Now, very small studies, in this case, only 17 people, but 59% show frank peripheral neuropathy with clear nerve damage. And other studies have looked at the same problem and seen some hints that there are immunological paradigms involved. Now, you can rule out autoimmune diseases like Sjogren's syndrome, but there are clear components of what is being termed disimmune mechanisms going on neurologically in these individuals. Lumbar punches, blood sampling, cranial MRIs, and CAT scans in 19 patients showed a kind of plasma cytokine storm response with some clear impairment of the blood-brain barrier. Something's breaking it down. And some hints at an inflammatory autoimmune response of some form going on in the brains of some of these individuals. Perhaps the one that's sparked the most conversation recently is a study that looked at the possibility that you're looking at an escalated aging syndrome, which may or may not be reversible, unclear at this time. So you look at the young brain, then the older brain. Some studies are now showing, based on a UK biobank brain imaging of 45,000 individuals pre-COVID and during the epidemic, a diminished gray matter in the frontal and temporal lobes of individuals who suffered COVID infection, not necessarily severe COVID, and not necessarily with diagnosed long COVID. Indeed, it looks like the loss of brain volume occurs regardless of the severity of the actual COVID disease, is focused around the olfactory bulb, which of course could explain the loss of smell and taste, and is in the same hippocampal area as is normally struck in Alzheimer's victims. This has also led to study asking, is the brain itself actually shrinking at a pace that is accelerated? The brain always shrinks with aging, but at about 0.2% a year. What has been seen is an escalation in the pace of brain shrinkage in some patients suffering from COVID. And it could actually represent a net 10-year escalation in the aging process of the brain. And so this, of course, leads to a whole set of concerns about whether or not we're looking at something that's really going to be lasting, and it's going to tax the individuals and tax healthcare systems for years, if not decades, into the future. The largest study looking at this problem was done by the Veterans Administration in the United States, looking at 154,000 veterans who have suffered COVID, diagnosed infection, compared to 5.8 million uninfected controls that have been through the system in the same time period, a roughly 10.5-month period in the United States. Looking at them a year later, and they see that survivors of COVID are 46% more likely to have been diagnosed with psychiatric disorders, including suicidal thoughts, depression, brain fog, sleep disturbances, and neurocognitive decline. The risk of brain fog, which again is a very mushy definition or diagnosis, is 80% higher than in controls. And that translates into 10.7 more cases per thousand in compared to uninfected populations, and a 343% higher risk of developing these disorders if the individual is actually hospitalized. Now, this seems to contradict the UK study, which indicated that severity of disease did not predict whether or not you would suffer neurocognitive disorders. In this case, a huge increase if you were in fact hospitalized with COVID. So we have a great deal to try and figure out about what is going on. And we hope that the kinds of very innovative tools that have been applied by our winners of this year's Michelson Prize will also be brought to bear in understanding and dealing with the future of these cognitive disorders and the tremendous possibility, horrible as it may be given the circumstances, that improving our understanding of these disorders could actually lead to breakthroughs in our understanding of how the brain ages, how dementia occurs, other perhaps Parkinson's and other disorders, and whether or not they can be treated through immunological means. So with that, I'd like to begin introducing you to this year's winners, an absolutely astonishing group of individuals who have had a real impact on their own work, their institution's work, and hopefully will have a big impact in the future of immunology writ large. First, I would like to introduce you to Dr. Rong Ma, who is currently at Emory University. She started out at City University of Hong Kong, then did her PhD at Emory University in the chemistry and mechanobiology department, and now is a postdoc in the laboratory of Khalid Salaita at Emory University. Her work is on mechanotechnology, nano, I assume, which measures and interprets mechanical forces that may be involved in the human immune system. She hopes to find a way to really measure and predict immune responses, and in particular in ways that could enhance the development of vaccines and of cancer therapy. Congratulations, Dr. Ma. I turn it over to you.
My name is Rong Ma. I'm a postdoctoral fellow at Emory University. I received the 2021 Michelson Prize for harnessing receptor mechanics as a marker for immunogenicity to isolate and identify potent T-cell receptors and recognized antigens. Curiosity has always been one of the biggest motivations that pushes me forward in research. My research focused on developing molecular sensors made of DNA, which can detect receptor forces on piconewton scale generated by cells when they are probing the surrounding environment. We and others in the field have found that the T-cell receptor employs mechanical forces as a part of its triggering mechanism in order to boost this specificity of antigen recognition. And by studying these forces, we found that the mechanical sampling of the antigen by the T cells is correlated with the T cell activation. So with this knowledge moving forward, we are trying to develop an assay to harness these mechanical forces transmitted through the receptors as a biomarker. This would be one of the very first attempts of harnessing the dynamic and biophysical nature of this antigen recognition process for potential cancer immunotherapy purposes. And in the long run, if this method is demonstrated to be effective for precisely identifying the potent T-cell receptor antigen bindings, we could use it as a strategy to generate personalized neoantigen cancer vaccine. As an early career researcher, the support from Michelson Prize will give me a lot of independence, as well as the peace of mind to focus solely on this research. And it allows me to pursue the high-risk, high-reward project of developing a novel assay for neoantigen and neoantigen-specific T-cell identification.
Congratulations, Dr. Ma. I have to ask you a couple of questions because it's so intriguing. Let's step back a second. When you say there are forces, and that there's some kind of a measurable force exerted by T cells, what exactly does that mean? Describe that.
So basically, the T cells have many TCR, T cell receptors on its surface. And what it does is it goes around and scan the surface of other cells. And when they encounter antigen, part of the TCR triggering mechanism is to employ this little tug on the antigen. And if this interaction is potent enough, it can withstand that force and the TCR get triggered. So a lot of groups have found this mechanism or found evidence that suggests this mechanism through different methods, but I think it's still pretty new for the world of immunology, which makes it really interesting for my proposal. Yeah.
Does it matter what kind of T cell?
Actually, the T cells that people have seen this phenomenon with, no matter it's with microscopy or single molecule for spectroscopy, they have a pretty wide span, I'd say. For example, the studies that we use, in our studies, we use naive CD8 positive T cells. Mostly they will, and we use cytotoxic T cells, which are the killer cells. But there are also groups that found this mechanism with CD4 cells, with Tregs, and even with B cells, the B cell receptor on the B cells itself. So it's still pretty new for the field, and it's still really evolving really fast right now.
Well, may the force be with you. I'm going to, and congratulations once again. I'm now going to turn to another one of your fellow winners, Camila Consiglio, who did all of her early work in Brazil. Yay, Brazil, at the molecular biology work at Federal University of Rio Grande do Sul in Porto Alegre, and then went on to do PhD work in immunology at Roswell Park Comprehensive Cancer Center, upstate New York, Buffalo. And now is a postdoc in Peter Broden's lab at the Karolinska in Sweden. So you're all over the place. You've seen much of the world, and you are working on a very interesting question, something that actually was kind of in reverse, a subject of my research when I was an immunologist at Berkeley's eons ago. I was interested in estrogen's effect on the immune system. You're looking at testosterone. And we know that you can go disease by disease, and you have different rates of infection and different rates of response based on gender, but it's very poorly understood. Dr. Consiglio is working with a unique cohort of individuals who are undergoing sex change transformation and taking measurements, I assume, throughout their treatment and after they have transitioned. So this sounds fascinating. Please tell us more.
I'm Camila Consiglio, and I'm a postdoctoral researcher at Karolinska Institutet. And I received the 2021 Michelson Prizes to understand how testosterone impacts the human immune system. I chose to become a scientist because I've always been very curious to understand the world around me. And so I chose specifically to go into the biomedical field to try to understand why people develop diseases and to try to find ways to better treat these diseases and prevent them. So my research is trying to understand how the immune system is different between men and women. And so we know that, for example, men tend to have more severe infections when compared to females. But on the flip side, women tend to have higher responses to vaccinations. So why is this? Why is it that women and men have these differences in immunity that can contribute to their susceptibility to different diseases, such as infectious diseases and autoimmunity? My research is trying to understand how a specific sex hormone, testosterone, that is different between males and females is impacting the immune system of humans. And so by trying to understand the impacts of testosterone in immunity, we can better understand how to prevent and treat infectious diseases and design optimal vaccine strategies that can fit one's biological sex. And so in my research, I'm combining highly advanced technologies that allow you to look at different components of the immune system all at the same time with computational methods. The Michelson Prizes will help me address a question that I've been interested in for a really long time, of how is it that males and females have differences in their immune responses. And so by understanding how biological sex impacts the immune function, we can now understand better how diseases are developing differently between males and females, and also optimize strategies to treat these infectious diseases, such as developing vaccine strategies that are fitting to one's biological sex.
That was terrific and very exciting work. You know, we know that there's a differential by gender in survival from infectious onslaughts in utero and in the neonatal period. Males are much more vulnerable and are far more likely to die in early life. And this is, of course, not just humans. This is, I think, most mammals. What does that tell you?
Yeah, that's an absolutely really important question for us to answer. So in the lab that I'm doing my postdoc in, we're interested in understanding the developing immune system in children. And this is one of the things that the MDs often tell us is when you know that there's a baby who's preterm and this baby is a baby boy, you're already more concerned for, you know, the effects that this baby boy might be at risk here. So increased infections and so on. And we still don't understand why this is happening. And so Sometimes we tend to think that these sex differences in infection severity are related to sex hormones, to testosterone, for example. But there's all these differences that happen before puberty, right? That could indicate maybe not a testosterone-mediated effect, but also sex chromosome effects. And so this question is really important to be figured out and to really discern the sex hormones and sex chromosome effects from one another, so we can really pin down what are these pathways in immunity. They're impacting individual immune cells, and how can we harness this for better strategies. And so I'm really excited that this is the direction that the Michelson Prize is allowing me to pursue to, take this to the next leap, I would say.
How long do you imagine you will need to be following these individuals?
So for the cohort that we're studying right now, we're studying them over the course of 1 year. And so we have baseline measurements before, how their immune system looked before they transitioned, before they receive sex hormones, and after 3 and 12 months after they receive sex hormone treatment. And so in this way, we're we're able to see how this short-term effect of hormone treatment, all the way to a very long term, I'd say 1 year, can affect their immune status.
Fascinating. Well, we're going to be eager to hear what you come up with. Thank you very much, and congratulations once again. And finally, I want to introduce you to Dr. Nicholas Wu, who is working on a whole range of issues related to how antibodies recognize their targets. He started off working at the University of Virginia in biochemistry, and completed his PhD at UCLA. And now, after a postdoctoral fellowship at the Scripps Research Institute, he is now an assistant professor at the University of Illinois in Urbana-Champaign. He's working on trying to interpret complex human antibody repertoire, the whole range of antibodies that may be involved in responding to a specific epitope, a target on whatever it is you're looking at. And he's using very sort of cutting edge, high throughput technology to get to the bottom of this and to antibody discovery. Let's take a look at his video.
My name is Nicholas Wu. I'm currently an assistant professor at the University of Illinois at Urbana-Champaign. Scientists are still very far from comprehending the full complexity of immunology. My major research direction is to understand the molecular interaction between antibodies and viruses. The research outcome will not only advance the fundamental understanding of immunology, but also facilitate the development of next-generation vaccines. So I really enjoy exploring novel ideas to push the boundary of knowledge. I also want to have a positive impact on the society. And being a scientist allows me to do both. I received the 2021 Michelson Prizes for my work on systematic identification of antibody epitope. With the recognition of the Michelson Prize, I feel more comfortable to explore high-risk, high-reward projects, both intellectually and financially. Data from these projects will potentially determine the research directions of my lab in the next decade. In addition, the Michelson Prize will help me establish international reputation, which is critical for my career advancement as a junior scientist.
Congratulations.
Thank you.
I think probably at this point, the best known epitopes in the world that the whole world imagination knows about are the spike protein epitopes on the COVID vaccine, the SARS-CoV-2.
Absolutely, yep.
And the various variants that arise where you can see that the virus is actively mutating to get around that immune response, the antibodies coming into those epitopes. How could your work fit in with that picture and perhaps help in our understanding of how escape mutations arise and how the immune system recognizes those escapes?
Yes, this is definitely an important question. What we have been looking into is to understand how antibodies target things, right? And then this spike protein is definitely like something that we are actively looking at. what we're trying to do is to understand how the antibody sequence. So every antibody has a sequence. They're just protein. So they have a sequence, and based on the sequence, can we predict where it binds to? So if we can, then it will allow us to develop vaccines against all these kind of variants. Because if we know what the variant is like, I mean, we have the sequence, and then if we can predict what can bind to those sequence, then it will help improve our vaccine design. We can specifically elicit some types of antibodies that can target the variants. Yeah, I mean, I would say this is still very early to say whether we can do it, but I do believe we are able to do that at some point.
Well, you know, it's interesting because one of the techniques that the invading world uses, whether it's microbes or cancer cells or whatever, is to basically sugarcoat themselves, wrap themselves in glycoproteins so that they are hiding. They put a coating over themselves so that our immune system doesn't recognize what you're talking about, the epitopes sticking out. Are you able to get to the bottom of any of that?
Yeah, I mean, if you look at I mean, some viruses have more sugar coating than others. For example, HIV is like so heavily coated by sugars. But our bodies are still able to generate antibodies against HIV. So if you look at the antibody response against HIV versus other viruses, we are seeing some distinct, like some sequence features on the antibodies that allow them to penetrate the sugar coat. So that's also something that we're trying to look at to understand how our body can get around that kind of hiding mechanism to target the virus. Yeah, I mean, this is definitely an exciting and ongoing active research direction.
So it's partly about epitope presentation and partly about epitope recognition.
Yes, that's correct.
Very different pieces.
Yeah, they're different pieces, but they're kind of related. So,
well, congratulations, Dr. Wu. It's really
Thank you.
Very exciting. And of course, we all want to see the results of your work. In fact, we all want to see the results of all of your work, because these are all very interesting projects that will have big implications going down the road. You know, I've said before that these are very competitive prizes, and they're ones that are much sought after. I think it may sometimes escape recognition that they have a big impact on how people go through the rest of their careers and what happens thereafter. I'd like to turn this now back to Dr. Wayne Koff, and perhaps, Wayne, you can say just a little something about what we know about how the Michelson Prize has affected the career trajectories of individuals who have been fortunate to receive the award in one of the prior rounds.
Well, thanks again, Laurie, and it's really wonderful to be here and to see each one of the videos, which I actually hadn't seen before, and see how well these individuals are going to go ahead. This is another class, if you will, of the Michelson Prize winners. I think we have 11 winners now in total over the 4 years. And we've already seen with the number of the people who received the initial prizes, a giant leap ahead in terms of the advancement in their own careers. One of the individuals, Ansa Sapathi at Stanford, received a job at Stanford shortly after the prize, has started a biotech company, has received NIH grants, etc. Another individual, Laura McKay, at the University of Melbourne, has also received a number of awards, has received internal and external support, and is doing extremely well in terms of the numbers of the publications in really high-tiered kinds of journals. We've seen this across the board for all of the winners, And I expect we're going to see the same for Nicholas and Rong and Camila. So we are proud of all of you. I think as you said in the beginning, we are just about to open up the portal of the next round of the competition of the Michelson Prizes, and we're looking for the best and the brightest out there. It's one of the best things that the Human Vaccines Project has done since the beginning. You know, obviously, our mission is to make an AI model of the human immune system, you know, which is a long way off. However, these kinds of individuals who we see, you know, here and who we've awarded in the past are key to the success, frankly, of us achieving our mission. And when we do achieve our mission, it's going to transform human health. And so I'm really proud of all of you, and you too, Laurie. You've been with us since the beginning, and it's been quite a ride. And we hope, you know, we'll have you back a year from now when we announce the next round of Michelson Prize winners.
Well, and you know, we have a little time, and I want to do something we've never done before. I want to ask Nicholas, Camila, and Rong to unmute and put your video back on. And what I want to do is ask each of you if you have a question for one of the other winners, something you'd like to know about their plan and their work. I'm going to start with you, Rong. You can pick who you would like to ask your question to.
Yeah.
Unmute. Yeah.
That's very exciting. So I actually was very interested in what Camila was talking about her research, the gender difference that she sees in the immune response. And it's just kind of resonate with what we have noticed in terms of the mechanobiology of these cells, no matter it's immune cells or platelets. Whenever I vaguely remember some of the data that one of our lab member collected a few years back. So when she put the cells on our tension probes and observe how active they are when there's a ligand for the receptors to recognize, they realized that there's actually a difference in male and female. Although we were using mouse, but there was difference. And it's just like kind of struck to me that her research there could be molecular mechanism behind this, and there could be a mechanobiology that is coupled with the molecular, the chemical part of it behind this phenomenon. And I thought that was a really interesting direction, and maybe we should discuss more about it.
That's beautiful. Yeah, I mean, this is the crazy thing and the most exciting part about being one of the winners here. You get to meet people that are doing such awesome science in a completely unrelated field, and yet we find a connection, right? Yeah. And so I didn't know about this, and I would even ask you back, you know, do you know what exactly was different between them? Do you think it's in terms of their cell membrane composition? Maybe it's the type of lipids that are in there, or, you know.
I'm not sure it's about the membrane fluidity of the cell membrane, but I think it's likely it's linked to the cytoskeleton dynamics inside the cells. And these cytoskeleton dynamics are actually regulated by a lot of other proteins inside the cells, which can be affected by, for example, hormones and some other factors, for example, the things that cells secrete if they're inflamed, or for example, we also have data that shows that T cells, specific T cells treated with serum that contained adipocyte before, which means they're like fat tissues, they are a lot less mechanically active, which partially explains, or at least resonate with why like people have these issues, might have a weakened immune system. So these are really interesting questions that we are very, very excited to pursue. And
I can't wait to talk. I mean, for the average person listening to us right now, let me translate a little bit. You're actually suggesting that the differences based on gender of the individual that these cells are inside of could actually be profoundly different. Cells that are not part of a gender-specific system, organ system. They're not of the genitalia, they're not breast cells or what have you. They're just that the actual structure of the cells might be different based on which hormones are circulating. That's fascinating. Nicholas, do you have a question that you'd like to ask one of the other winners?
Yes, I also found this gender difference issue quite fascinating. So like when we I'm not sure if it also apply to innate immune response, but like when we study tissue culture, we use a lot of tissue culture to study viral infection. Do you think gender, like this kind of gender difference, is also happen in innate immune response, so like a tissue, in a cell culture level? Since like when we're using the cells, so the like different cells, they're from different people with different genders, right? So I'm wondering if that actually creates some sort of artifact, or I wouldn't say artifact, but like some
Absolutely.
Just so that our listeners or viewers follow this, because this is a big deal. You're suggesting it's possible that a lot of immunology research might be flawed because it's failed to account for what the gender was of the individual animal or human that the cells came from. What do you think, Camila?
No, I think it's a really good point to make. I think until I was doing my PhD, it wasn't necessary for grants at the NIH, as I believe, to include why you're, or how do I put this, which biological sex you're studying. You weren't really necessarily needing to justify. And I think starting 2015, 2016, you had to justify, why are you using females or male mice or subjects to, understand such biological concepts. And this is so important because, coming down to your question too about innate immunity, it's been shown that biological sex impacts immunity at different levels. So you look at innate immune cells, you have changes in macrophages, like phagocytosis is different between the sexes, and K cell function. And then you look at adaptive immune function, T cells, B cells, all of these are all being affected by sex hormones and by chromosomal sex as well. And so if you take into account just a Jurkat cell that might come from a male, I'm not actually sure, then you're completely biasing yourself, right? So we need to take into account that these biological factors matter, and really take this into consideration for our studies, for our conclusions, and for next directions.
Well,
That's very
Nicholas, I'd like to throw it to you on that one, because we've gone pretty far down the path of research in immunology over the last several decades before people really start saying, wait a second, what's the age of the donor from which you've derived the cells that you're studying, and how does age affect this immune response? Now you're going to ask, what's the gender?
Exactly.
How does that affect the immune response? Do you think in your epitope system, you could factor for these issues?
Yes, that's actually one of the areas I'm really interested in looking into, because our immune system actually kind of like evolve over time as we are accumulating more, as we are accumulating immune history. For example, it has been shown that people with different influenza infection history will respond to flu infection very differently. It depends on the types of flu, like the actual strain of the flu. We hope to, like, once we have a system built up that we can predict what antibodies bind to, then we can look into individuals' repertoire of antibodies and see and try to predict if they would respond differently to different flu infection or like different virus infection. And then, of course, this is correlating to age, because as people age, as I said, the immune repertoire evolve. We're not sure what we will find, but that's certainly something that we should be able to look at.
Well, and of course, as in my remarks, I was saying how difficult it is for us right now to understand long COVID because of the epidemiology of it. Are we looking at an actual pathogenesis resulting from the virus itself or from the immune response to the virus? Or are we looking at general social factors of social isolation and depression induced by what we've all been through, living in our homes forever, getting sick of our offices and our families and our squished together lives? And I can imagine that you're going to have the same problem looking at gender because, or age, because people's lifestyles are different. The nature of their external exposures, their workplaces, how much they are the primary caregiver of children and exposed to all the antigens carried by their children from school and so on, what kind of a workplace they are in, what they're exposed to in that workplace. Suddenly you realize that correlation and causation becomes a tricky problem across the board. I wonder if any of you have any thoughts about that.
Well, I'll jump in, Laurie. I think that's absolutely true. And I think we, have seen it, frankly, over the last 18 months. We have, the elderly, of course, have the most of the morbidity and the mortality in this, in this period. But there are a number of elderly individuals who have done extremely well with this virus and who've not gotten sick. And there is a hypothesis out there that we and others have made that the baseline is predictive of the outcome, and we don't understand the baseline. And so one of the things and one of the interest that we have certainly in the Human Vaccines Project, we've done a lot of work in the B cell and the T cell the repertoire areas, and sequencing of the BCRs and the TCRs. But as Nicholas has pointed out, that we're a long way away from the map of what the ligands are of each B cell receptor and each T cell receptor. But I actually see that as feasible over over time. The exponential increase in the tools that we have and the sensitivity of the tools. I mean, just remember a decade ago, we weren't involved in any single cell analysis at all and such. And we didn't have the capacity, the capacities with AI and machine learning now to begin to have models of systems in ways that we have now. So I think we're going to solve these problems. It's going to take us a little bit of time, and it's going to be a long and winding road, as we know. But as Nicholas pointed out, I mean, and the influenza virus is a perfect case. Each one of us has an immunologic imprint, and we get that imprint generally in the initial 18 months of life, and we don't understand anything about it yet. And if we did understand it, it would give us a whole new set of ideas about how to make a universal influenza vaccine, and similarly how to make a universal vaccine against the coronaviruses. So I guess what I'm trying to say here is that the human immune system is at the core of everything in terms of infectious and non-communicable diseases. And we're learning more and more about it every day. And again, we have examples here of three of the up and coming, leading, researchers who are going to trailblaze here for the decades to come. So I'm not sure I answered your question, but I think this is a really exciting time for all of us. And hopefully, as you know, Laurie, as we've done in the past, hopefully this is the last of the Zoom award ceremonies for us, and we can get back into in-person meetings and the dinners and the award ceremonies that we've done.
And a glass of champagne to each of you.
And we bring in we bring in all of the winners of the years into those meetings, and we've already have seen a number of the collaborations that have just occurred over the dinners. So we look forward to in welcoming you all back at our next award ceremony a year from now, hopefully in person, hopefully at a nice place, and hopefully with a nice meal.
And I would just to tell the audience so that people really understand the significance of what Gary and Alia Michelson have so generously done with this award is that if you look at the data now on grants out of the NIH, you can see that they are heavily skewed to longtime grant recipients. It's very hard for a young scientist to break through all those barriers and get their share of the pie to do their research because it's so dominated, so heavily skewed towards firmly established, tenured researchers at major research institutions. And so the trick of what happens with the Michelson Prize is it gives you a little nudge, just a little edge to get you closer to those much older and more established scientists so that you're on a grant pathway that will carry you through to your professional career and allow you to build your own laboratory and move into this space on a permanent level. And I hope that you find that the little nudge that we're giving you today will help you in that direction. And it's for the good of all of us that are watching you because we want to see the results of your work. I thank everybody involved in the Michelson Prize, particularly Gary Michelson himself and Wayne Koff and all the good folks at the Human Vaccines Project for allowing us to share the joy with you and to hear about your work. And to all of you, again, who are watching and are young scientists yourselves, the deadline, it opens up April 1st for your applications for the next round of the Michelson Prizes. Thanks, everybody. I think we're at our conclusion. And again, happy smiles go off and I guess privately have your glass of champagne.
Thanks, Laurie. And thanks
Laurie.
to all of you.
Thank you so much.
Thank you. Thank you.
Goodbye to all.
All right.